We will begin exploring one of the most remarkable systems in organismal biology, the immune system. This system is constantly working in the background, defending the body against a wide range of pathogens while still recognizing and protecting our own cells. What makes it especially fascinating is how it blends simple physical barriers with highly specialized cellular interactions. As we move through this lecture, we will connect those concepts to real biological processes that help organisms survive in dynamic environments. This gives us a solid starting point before we dive into the specific components and the mechanisms that will make immunity possible. As we begin looking at the immune system, it helps to think of it as a coordinated network rather than a single structure. Every part of it, from physical barriers to specialized cells, plays a role in protecting the body from infections. What makes this system exciting to study in organismal biology is that it represents one of the most ancient and diverse defense strategies across animals. You will see that immunity blends molecular precision with whole body responses, and both levels work together to keep organisms alive. That gives us the foundation we need before moving deeper into other major components. To understand immunity, we need to know what the system is designed to stop. Pathogens are the biological agents that cause disease, and they come in many different forms, including bacteria, viruses, fungi, protists, and parasitic organisms. Each group evolved unique ways of entering and exploiting a host, which is why the immune system needs multiple layers of defense. The diversity of pathogens also explains why immunity is never a single reaction, but instead a series of responses. The immune system does not begin with white blood cells. It starts with the body's surface, when simple but powerful barriers prevent most pathogens from ever getting inside. Skin keeps microbes out by drying them out and creating an acidic environment, while beneficial microbes complete their invaders for space and nutrients. Inside the body, mucus traps particles, cilia push debris out of the respiratory tract, and tears and urine wash away potential threats. Even the stomach provides a defense with its low pH. These barriers act continuously, forming an essential shield that reduces the burden on deeper immune mechanisms. Once pathogens breach those barriers, the innate immune cells respond. Mast cells are among the first to react, especially during injury or infection. They release histamines and heparin, which expand nearby blood vessels and draw more immune cells to the site. This same sensitivity makes mast cells important for wound healing, but also responsible for allergic reactions when the system misinterprets a harmless substance. Their position in connective tissues and mucous membranes allows them to serve as an early alarm that activates the rest of the immune response. Macrophages are one of the most versatile cell types in immunity. They migrate from blood vessels into tissues where they engulf pathogens and damage cells through phagocytosis. Beyond cleanup, they signal to other immune cells to help coordinate both the innate and adaptive response. Their ability to recognize threats and alert the rest of the system makes them connectors between early defenses and long-term immunity. As we move forward, you will see how other cell types build on this foundation to create a complete response to an infection. Natural killer cells gives the innate immune system a surprising amount of precision. Instead of waiting for a pathogen to fully identified, natural killer cells recognize when a host cell is being abnormal, such as when it is infected by a virus or becoming cancerous. They act quickly by triggering apoptosis, allowing the organism to eliminate dangerous cells before the infection spreads. This rapid surveillance system is essential for keeping tissues healthy. Their ability to patrol both blood and tissue creates an early line of defense that bridges the innate and adaptive cells. Dendritic cells act like the translators of the immune system. They first behave like innate cells, sampling their environment and capturing pathogens. Then they take on an adaptive role by processing those antigens and presenting them on T cells. Because of that dual identity, they are essential to activating long-term immunity. They act heavily concentrated in epithelial cells like the skin and digestive tracts, places where pathogens are most likely to enter. Once activated, they migrate to lymph nodes to begin the adaptive response. Monocytes circulate through the bloodstream in an immature form, almost like reservoir troops. When they receive signals of inflammation and leave the blood and enter tissues, they are differentiated into macrophages or dendritic cells. That flexibility helps the immune system scale its response depending on the severity of the infection. The spleen serves as an important storage site for monocytes, allowing a rapid release when the body detects injury. This connection between organs and circulating cells shows how immunity operates as a coordinated network rather than in isolated parts. Neutrophils are the most abundant leukocytes and are usually the first to arrive at an infection site. They release antimicrobial components that damage bacteria and fungi, clearing out threats quickly. However, their aggression comes at a cost, sometimes causing collateral tissue damage. Their short lifespan reflects the intense work they perform in an early stages of inflammation. As they act, they also help recruit other immune cells to build on more sustained response, creating an important handoff to macrophages and lymphocytes. Basophils add another layer to the inflammatory response, especially in defenses against parasites. They release histamine, which expand blood vessels and help immune cells reach infected tissues. That activity often overlaps with mast cells, but basophils circulate in the blood before migrating to the tissues. They are also key players in allergic reactions, where the immune system reacts strongly to antigens that are normally harmless. This sensitivity highlights the balancing act the immune system must maintain between protection and overreaction. Eosinophils specialize in dealing with larger parasites and cannot be phagocytosed easily. They release enzymes and toxic proteins that can damage those organisms, although the same chemicals can injure host tissues when released in excess. Their presence increases in allergic reactions and parasitic infections, making them useful diagnostic indicators. By circulating through the blood and entering tissues during inflammation, they add a target but powerful layer to the innate immune response. Interferons act as early warning signals during viral infections. When a cell becomes infected, it releases interferons that alert neighboring cells to heighten their antiviral defenses. This does not save the original cell, but it slows viral spreading through the tissues. Think of interferon as a broadcast message that forces nearby cells into a defensive state, buying time for the rest of the immune system. Their role highlights how immunity depends on communication as a direct pathogen destruction. Inflammation is one of the most familiar immune responses, but it is also one of the most coordinated. A small cut activates mast cells, which releases histamines that widen nearby capillaries. Neutrophils and monocytes exit the bloodstream and enter the tissues to begin phagocytosis. As monocytes mature into macrophages, they release chemical signals that amplify the inflammatory response. This cascade ensures that pathogens are contained and cleared out quickly. It also connects directly to the complement system, which we will look at next. The classic complement pathway begins when antibodies bind to the antigens on the surface of a pathogen. This binding event creates a signal that attracts the C1 complex, which attaches to the antibody antigen complex and activates the rest of the cascade. Once C1 is engaged, a series of complement proteins activates one another in a chain reaction that rapidly amplifies the immune response. As the cascade progresses, complement proteins tag the pathogen for phagocytosis, stimulate inflammation, and ultimately form a membrane attack complex that punches holes in the pathogen's membrane. This pathway shows how adaptive immunity and the innate immunity work together. Once antibodies form, the adaptive system initiates a powerful innate effector response. Adaptive immunity provides the specificity and memory that the innate system lacks. It activates when the innate response cannot fully control an infection, and it tailors its reaction to the specific antigens present. The adaptive system has two major branches. T cells drive the cell-mediated response, while B cells and antibodies drive the humoral response. Neither branch can activate on its own. They rely on signals and antigens present on the innate system. This interplay allows organisms to develop long-term protection and rapid responses upon future exposure. Antigen-presenting cells are the bridge between the innate and adaptive immunity. Their first job is to encounter, engulf, and digest pathogens on the tissues. Once they process those pathogens internally, they move the fragments to their surface and display them using MHC molecules. This display function is what allows T cells to recognize when a threat exists. Without antigen-presenting cells, the adaptive system would remain blind to infections, no matter how severe. An antigen is any molecule capable of triggering an immune response, but the key feature is its structure. Each antigen contains multiple epitopes, which are small specific regions recognized by immune receptors. These epitopes determine how the immune system identifies a pathogen and which cells participate in the response. When you look at an antigen, think of the molecule's landscape with several distinct features. T cells are at the core of the cell-mediated branch of adaptive immunity. They cannot recognize antigens on their own, so they depend on antigen-presenting cells to show them the process fragments that MHC molecules. Once activated, T cells differentiate into a specialized group. Helper T cells coordinate much of the immune response by activating B cells, macrophages, and other lymphocytes, while cytotoxic T cells directly destroy virus-infected or abnormal host cells. Regulatory T cells help keep the system in balance and prevent overreactions. Together, these subtypes ensure that the immune system responds with precision while also developing long-term memory for further exposure. The T cell receptor determines what each T cell can detect. Its variable region forms a unique binding site that matches a specific epitope present on an MHC molecule. When the receptor binds its matching epitope, the T cell becomes activated and begins its response. This interaction is highly selective, which ensures that the adaptive system reacts only to the correct threat. That structure of the TCR makes T cells incredibly powerful, but also dependent on the antigen-presenting process we saw earlier. B cells form the foundation of humoral immunity. Unlike T cells, they can recognize intact antigens directly on the surface of pathogens. When B cells encounter their matching antigen, they become activated and can differentiate into plasma cells. These plasma cells are responsible for producing antibodies in large quantities, spreading immunity throughout the body. B cells also form memory populations that allow for faster and stronger responses upon their later exposure. B cell receptors make this entire process possible. They are embedded in the cell's membrane and serve as antigen detectors. When a B cell receptor binds to an antigen, it triggers internal signaling that activates the B cell. Each receptor has a unique shape determined by genetic rearrangement, which creates enormous diversity across B cell population. Natural killer cells are part of the innate immune system, but they work with a level of precision that resembles adaptive immunity. Their primary job is to identify and eliminate cells that are behaving abnormally, such as those infected by viruses or beginning to transform into cancer cells. They carry out this task by inducing apoptosis, which prevents harmful cells from spreading infections to neighboring tissues. Natural killer cells act quickly, often before the adaptive system even begins to mobilize. Their speed makes them one of the body's most important early defense tools. Natural killer cells make their decisions by scanning for molecular signals on the surface of host cells. Healthy cells display specific markers that tell natural killer cells to leave the cell alone. When those markers are missing or reduced, which often happens when viruses interfere with normal cell function, natural killer cells interpret that as a dangerous sign. This ability to identify what is missing rather than what is present gives natural killer cells an advantage over pathogens. Mucosa-associated lymphoid tissue provides immune protection at the body's major entry points. These regions line the respiratory, digestive, and urogenital tracts, where exposure to external environments is constant. Within MALT, clusters of lymphocytes stand ready to respond to foreign particles that cross mucosal surfaces. Because so many pathogens enter through these routes, MALT is one of the busiest and most active components of the immune system. Its widespread distribution helps organisms respond quickly to local infections before they spread. Different regions of MALT serve unique processes. In the digestive tract, patches of immune tissue monitor food and microbial contents. In the respiratory tract, similar clusters guard against airborne pathogens. Although these tissues are scattered throughout the body, they work together as a coordinated network. Their constant surveillance ensures that the immune response begins at the earliest stages of pathogen entry. Memory B cells allow the adaptive immune response to improve with experience. After an infection is cleared, some B cells do not become plasma cells, but instead remain in the body as long-lived memory cells. They carry a precise record of the antigens they have encountered, so if the same pathogen returns, they respond much faster than during the primary exposure. This rapid activation produces large amounts of high-affinity antibodies and often prevent noticeable symptoms. Their presence is the biological foundation of long-term immunity and the principle behind how vaccines protect us over time. When the body's first encounters a new pathogen, the immune response is slower because the system is identifying the antigen and activating the appropriate B and T cells. This is called a primary response. The antibody production is moderate. During a second exposure, memory B cells and T cells activate almost immediately. The secondary response produces a much larger amount of antibodies and sustains it for a longer period of time. This presence shows us how the adaptive immunity improves with experience. Lymph is the clear fluid that travels through the lymphatic vessels and serves as a transport system for immune cells. As lymph flows through the body, it passes through lymph nodes where lymphocytes screen for foreign cells and antigens. This movement allows the immune system to monitor tissues without needing to be present everywhere all at once. When lymph enters a node through the afferent vessel, it encounters a dense population of T cells and B cells that can intercept pathogens. After filtering, the lymph exits through the afferent vessels and continues circulating. The spleen is the largest lymphatic organ and plays a vital role in filtering blood rather than lymph. It contains red pulp, where old or damaged red blood cells are removed, and white pulp, which houses lymphocytes that detect blood-borne pathogens. Because of its structure, the spleen acts as both a recycling center and an immune checkpoint. It ensures that the bloodstream remains clean and the immune cells react quickly to any microbes that manage to enter into circulation. Antibodies have a recognizable Y-shaped structure that reflects their dual function. The tip of the Y containing variable regions which differ from one antibody to another and allows them to bind very specific epitopes to an antigen. The lower portion of the molecule, called the constant region, is the part that determines the antibody class and dictates how it will behave in the immune system. This region interacts with immune cells, activates complement proteins, and influences where the antibody travels to the body. The hinge between the arms gives antibodies flexibility, making it easier for them to bind antigens to a different position. IgA plays a major protective role at the body's mucosal surfaces. You find it in saliva, mucus, tears, and breast milk, which tells us how important it is for guarding entry points where pathogens first attempt to colonize. Its ability to neutralize microbes before they penetrate tissues makes it a front-line antibody. In infants, maternal IgA provides early protection while their own immune systems develop. This class shows how antibodies can be specialized for specific environments rather than functioning in the bloodstream alone. IgD is the larger abundant antibody classes, but it plays an essential role in early B cell function. You can think of it as part of the sensor system that helps B cells recognize antigens for the first time. IgD sits on the surface of the developing B cells, and when it binds to its antigen, it helps trigger an activation process that allows the cell to mature and respond. While only a small amount of IgD circulates in the blood, it also contributes to activating basophils and mast cells, connecting it to the inflammatory response. IgE is the antibody class best known for its role in allergic reactions, but its original evolutionary purpose was to protect the body from parasitic worms. IgE binds tightly to mast cells and basophils, essentially priming them to release histamine when they encounter a matching antigen. When the antigen is a harmless substance like pollen or food proteins, the response becomes an allergy. Even though IgE circulates in very low amounts, its effects are very powerful because a single activation event can trigger widespread inflammation. This sensitivity makes IgE both an important defender and a major contributor to hypersensitivity reactions. IgE is the dominant antibody in the bloodstream and tissues, which make it the primary defender against pathogens circulating throughout the body. It is highly effective at neutralizing viruses and bacteria, tagging them for phagocytosis and activating complement proteins that help destroy microbial surfaces. One of the most important features of IgG is the ability to cross the placenta, giving developing fetuses passive immunity before birth. It is also the main antibody produced during secondary immune responses, which means it becomes more abundant and more effective after repeated exposures to the same pathogen. IgM is the earliest antibody to appear when the immune system encounters a new pathogen. Its pentamer structure gives 10 antigen binding sites, which makes it very effective at clumping pathogens together and activating the complement system. Because of its size, IgM stays largely in the bloodstream, where it helps contain infections before other antibodies classes develop. Clinically, the presence of IgM is a useful indicator of a recent or active infection, since it appears before IgG levels rise. IgM shows how the immune system uses rapid, broad responses first and then shifts to more specialized antibodies as the adaptive response develops. Antibodies carry out several different defense roles once they bind to a pathogen. They can neutralize microbes by blocking structures the pathogen needs to infect cells. They also tag pathogens so phagocytes recognize and consume them more efficiently, a process called opsonization. Some antibodies activate complement proteins that help destroy microbial membranes. These functions allows antibodies to operate effectively in many environments throughout the body. Each mechanism contributes to coordinated efforts that keep infections contained and eliminates threats before they spread. Affinity refers to how tightly a single antigen binds to its antibody attached to its epitope. Avidity describes the overall strength of the binding when multiple sites are interact at once, which is especially important for antibodies with several binding sites, such as IgM. Cross-reactivity occurs when an antibody designed for one epitope can also bind to a similar structure in a different molecule. This can sometimes help broaden protection, but it can also contribute to autoimmune reactions when the similarities are too close. Immunodeficiencies occur when the immune system cannot respond effectively to infections. Some forms are inherited, affecting key steps in developing or signaling. Others are acquired, such as those caused by viral infections, malnutrition, or medical treatments that suppress immunity. When the system is weakened, even minor pathogens can cause severe illness. Studying immunodeficiencies helps us illustrate how each component of the immune system contributes to overall health. Hypersensitivity reactions occurs when the immune system responds too strongly to a harmless antigen. The most familiar example is an allergy where IgE-mediated responses trigger mast cells to release histamine within minutes to exposures. These reactions can range from mild irritation to life-threatening anaphylaxis. Autoimmunity occurs when the immune system can no longer distinguish self from non-self and begins attacking the body's own tissues. This failure of the self-tolerance can involve autoantibodies, autoreactive T cells, or both, leading to chronic inflammation. Lupus is one of the most well-known autoimmune diseases and a good example of how widespread the effects can be, since it targets multiple organs and produces a wide range of symptoms. Its development reflects a combination of genetic risk factors, environmental triggers, and breakdown of its immune regulation. Autoimmunity shows how a system designed to protect the body can also cause significant harm when its control mechanisms fail.